and then, yeah, okay, awesome.
So hi everybody, welcome to Machine Learning and Friends Lunch. We are delighted to present Professor Marinka Zitnik today, who is joining us from Harvard University. Dr. Zitnik's work is on the application of machine learning to biomedical data and is driven by a vision of artificial intelligence techniques that support clinical work and accelerate scientific inquiry. Today, Professor Zitnik will be talking about advances in graph neural networks and their medical applications. So if everyone can unmute themselves real quick so we can welcome the speaker. Thank you so much for joining us today. And then I just wanted to let you know, if you have any questions, you can raise your hand during kind of the pauses in the talk and I'll call on you. If you have an extremely urgent question in the middle of the talk, feel free to raise your hand and DM me in the chat or something, and we'll figure out how to interrupt. But great, thank you so much. Thank you, Professor Zitnik. So you can get started.
Hello, everyone. I'm delighted to be here today, and thank you, Nika, for inviting me as well as for giving such a nice introduction. I'm sharing my screen, so I hope you can see my first slide. If not, let me know. The title of today's talk is Actionable Machine Learning for for development of safe and effective therapeutics. So let's begin. Science crucially depends on existence of scientific instruments. And in the past, those scientific instruments were primarily physical devices, such as microscopes that have facilitated discoveries. However, nowadays, when we are in the era of modern data-intensive sciences, and we need a new kind of instruments that I envision will be in vital ways optimized for knowledge discovery from data. Those instruments will not be physical devices anymore, but can also be virtual devices, so algorithms and machine learning tools that we are developing. So this is really a great vision that is shared by many in this field, but it's still very challenging to realize this vision in practice in scientific arena today. So for that, there are several challenges that prevent us from realizing this vision, and I will mention two broad challenges today. And so the first is that for most of the scientific questions, in particular questions that arise in biology and medicine, there is no single data type that is sufficient to explain and model the complex phenomena that we are encountering, such as a disease or the way the drug would work in human body. That means that there is no single large data set that one can work with, but there are multiple related relevant data sets, and one will need then to integrate this heterogeneous, often confounded data that span from molecules all the way up to populations and society levels. That is challenging because data is multi-scale. It is describing behavior of molecules, individuals, entire populations. It is heterogeneous in the sense that it comes in the form of experimental readouts, curated annotations, or self-reported data. And it might be confounded because it can be generated by a variety of different technologies, measurement platform, or collected at different institutions, different geographic regions. Second important challenge that we need to address is that machine learning models need to be able to generate actionable predictions that lend themselves directly to testable hypotheses in downstream experiments in the wet lab or in clinical setting. So that means that we need to find ways to provide predictions that are not only accurate, but also can be interpreted meaningfully. And so we develop strategies to take black boxes that might be very accurate that we have today and make them more open boxes that allow us for meaningful explanations and as well as interpretations of predictions. In my group, we are using the power of networks or graph as a general language to bring together these heterogeneous data sets that we encounter in biology and medicine. And so networks are a general language that can serve us to represent very rich, heterogeneous, interconnected data at the level of populations, where we can think of networks of individuals that are connected based on friendship or familiar relationships or genomic similarity, similarity of genomic profiles, going down to the level of diseases within a single person, within an individual, and the way a variety of different diseases manifest themselves within an individual, and all the way down to the level of cells and molecules where a variety of cellular components and molecules interact with each other. And so networks are appealing because they allow us to bring together these data at these different scales, and also to obtain a unified mathematical representation that can represent this rich multimodal data set. Of course, very natural question to ask once we have these rich network representations of our of models that we are trying to of problems that we are trying to model. A natural question to ask: how can we train powerful deep learning algorithms on these networks, on graphs? And so the question then becomes this: well, we need to find a way to be essentially able to perform convolutions on these rich multimodal graph inputs, and then perform a series of nonlinear transformations that would allow us to finally arrive at predictions about various properties of nodes and entities that appear in our input graphs, or various kinds of novel relationship or associations between entities in the graphs. So networks or graphs are powerful data representation, but they are challenging to work with for classic deep models. So what I mean by that is the following. Classic deep models were primarily designed for grids or simple sequences. So convolutional neural networks or a variety of architectures in that field were designed for fixed size images. You can think of an image as a graph where every pixel would be represented by a node that has 4 neighbors, pixel above, below, to the left and right. And we might have multiple grids like that if each corresponding to a different channel. But this representation gives rise to regular grid, regular graph. On the other hand, there are a variety of very powerful deep models and algorithms designed for sequential data, such as natural languages or general sequences, including protein sequences. And in many cases, we can think of those sequences as simple chains or graphs where every token would be represented by a node. And that node in the graph has 2 neighbors, which is token to the left and token to the right, minus the first and the last token, of course. So we have seen that these models have brought extraordinary gains in a variety of fields, including computer vision, natural language processing, speech, and robotics, but are really unable to consider interactions, the essence of biomedical networks, because networks or graphs that we see in real world don't look like grids. They don't look like simple sequences. They have much more complex, richer topology. And so one cannot simply apply classic deep models to graphs, not only because they have richer topology, but also because certain kinds of invariances that can be leveraged in images and sequences are simply not valid in the context of graphs. So a few years ago, that gave rise to a powerful new class of neural networks that are known today as graph neural networks. So the key technical concept in graph neural network is that of network embeddings or graph embeddings or node embeddings, where intuitively you can think of this problem as following. We have as input a graph with some nodes and edges. In this case, it's just a simple graph with just 1 edge per every node and no particular types of edges, but in reality, graphs can be much more complex. And so graph neural network specifies a deep transformation function f that maps every node in that input graph to a compact vector embedding, such that 2 nodes in the input graph that have similar network topology, that have similar local neighborhood, are represented by points in the embedding space that are close together in that vector space that is learned, right? So why is that important? That is important because the embedding space is optimized in such a way that it reflects topology of the input graph. And so the key question then becomes how to specify that transformation function f that will map nodes from the input graph to points in the embedding space, such that performing linear algebra operations in the embedding space will be reflective of topological similarity of nodes in the input graph. And so typically the intuitive idea is that we specify those functions F in such a way that they map nodes to these d-dimensional embeddings, such that nodes U and V, if they are considered to have similar network neighborhoods, are embedded close together. Now, there are a variety of different architectures and different graph neural network models that operationalize different notions of what it means that two nodes have similar network neighborhood. Depending on that, then one can get different kinds of embeddings. So at broad scale, the implication of graph neural networks is that we can now apply deep learning much more broadly, not only to medical images, not only to biological sequences, but to any network that we encounter in biology or medicine. And so this creates exciting new opportunities in the area of biology and medicine. And so to this end, I will talk today about 3 vignettes. In particular, first, I will focus on the use of graph neural networks for predicting safer medicines and identifying safe drug combinations. Second, I will talk about graph neural networks for development of effective medicines and drug repurposing. And finally, I will conclude with an overview of how graph neural networks allow us to tackle very hard challenges where we need to reason about never before seen phenomena in emerging diseases. And along the way, I will also highlight some specific new technical contributions that we made in the group as a result of tackling these exciting new problems at the frontier of biology and medicine. Okay, so let's start with the first topic, which is that of drug safety and drug combinations. So the context and motivation for this project is the following. There is a notion known as polytherapy or polypharmacy that is used to refer a setting when patients take multiple drugs at the same time to treat complex disease or many coexisting conditions. In fact, that is very common. 46% of people, so nearly half of people over 65 years, take more than 5 prescription drugs at the same time. It's not uncommon to encounter patients that take more than 20 drugs at the same time to treat heart disease, depression, or cancer. And so the problem with that is that individuals that take many drugs at the same time are at much higher risk for developing unwanted adverse events. And around 50% of the US population is affected by those unwanted adverse events of drugs, which represents a problem both for individuals as well as for healthcare system, because annual costs in treating side effects are skyrocketing. And so that is a huge problem that cannot be tackled experimentally simply through clinical trials, because the sheer number of possible combinations of drugs is humongous. Imagine taking 5,000 approved drugs in the US system, and just the number of combinations of 2 drugs is 5,000 just 2. If you think of combinations of 3 drugs, it's 5,000 just 3. So combinations of 5 drugs would require us to experimentally model 5,000 just 2 combinations and do 5,000 just 2 tests in wet laboratory. So that is certainly not feasible. And that's why computational methods are needed to a priori define and identify those combinations of drugs that might be unsafe for patients so that they are not prescribed to real-world patients. I said that when patients take multiple drugs at the same time, they're at much higher risk for adverse events. Why is that the case? That is the case because the drugs do not work independently of each other, but there are some kind of interactions between drugs. So let's look at this, look at that in this particular hypothetical setting. Imagine we have a system with 2 drugs. We have blue pill and we have red pill. And when a patient takes blue pill alone, the desired therapeutic effect is achieved, no side effect occurs. When patient takes red pill alone, everything is okay, no side effects. However, when patient takes red and blue pill together, then with some probability, the patient will experience side effect. And so that is unexpected. Why? Because if the 2 drugs, blue and red one, would act independently of each other, then we would not expect any side effect here. Clearly, there is something going on that when 2 drugs are taken together, we see effects that go beyond just additive effects of individual drugs in the combination. So this will be our key test that we'll try to model today. Our learning test is, we want to have a model that will take as input a particular combination of drugs, and it will produce as output the probability that that combination of drug leads to a particular side effect, such as headache or insomnia, or a certain type of tumors. And those side effects cannot be attributed to either drug alone in the combination, but are really due to emergent interactions between drugs. So to model this problem, we constructed a large polypharmacy knowledge graph. And so that knowledge graph had information about drugs. So every triangle node here is an approved drug in the US. 2 drugs are connected by an edge of type R1. If there is known information that when patients take the drugs encoded by the endpoint triangles together, then a specific type of side effect, R1, occurs with certain probability in patients. And that side effect is due to that pair of drugs acting together. So in addition to that network of known interactions between drugs, we integrate in the knowledge graph another mode of information, that on proteins, that represents how drugs affect human body in the sense of what are the proteins that those drugs target and how those effects propagate in molecular networks. And so that gives rise to the second mode of information represented by orange circles encoding human proteins and interactions between those proteins in cells. So polypharmacy network is then a knowledge graph with 2 types of entities, proteins and drugs, and a large number of edge types that indicate various kinds of side effects, as well as drug target edges and protein-protein edges. So the idea then will be that we want to take the overview of the approach, or the key idea is that we take the polypharmacy knowledge graph as input, and we want to specify our graph neural network that will map each and every node in that graph as in the embedding space and represent it as a particular point in that space, such that the topology of such that embedding space will reflect topology of the input graph. And so the key idea to generate embeddings is that on examining local interaction neighborhoods around each and every node in the graph, and then generating embeddings based on the structures and topology that can be observed in those local network neighborhoods. So for each node in the graph, say node V, which might be the drug node, we examine certain neighborhoods of certain size. What is shown here is 2-hop neighborhood of node V. And in that 2-hop neighborhood of node V, we see that V interacts with a variety of other drug nodes, a variety of other triangles to different kinds of interactions, R2, R3, R1, and in turn, then their first order neighbors of V then have their own interacting nodes. So the idea here is to determine nodes computation graphs. For every node, we examine this local network neighborhood, and we get a computation graph for each edge type. So for R1 separately, for R2, for R3, and so on. And so once those computation graphs are obtained, then the graph neural network function F will learn how to effectively propagate and then transform information or neural messages along edges of those computation graphs. And so the process of that propagation and transformation of neural messages will yield embeddings. So very simply, and this is really a basic model that was state of the art in 2018 when this work was done, but now there are many, many more advanced algorithms that were developed based of this work in 2018. So mathematically, the equations are relatively simple. So our multi-relational graph encoder in this approach has the following structure. So embeddings for every node has an embedding at each layer. So initial embeddings for nodes are site features that we might have about nodes, or if no site features are available, then we simply use one-hot encoding vectors. So H sub V would indicate an embedding for node V at layer 0, which would be just initial one-hot encoding vector or feature vector for node V. Then at each layer of this graph neural network, the following operation is performed. To get at an embedding for node V at layer K, we perform the following update. So node V will look at, will construct this computation graph that I have mentioned. We go through every possible edge type. For every edge type, we check what are the neighbors of node V in that edge type. So for a given neighbor U, what we would then take the embedding of node U from the previous layer. This is the neural message from that node U would send to node V. And those messages are then aggregated through a trainable weight matrix W, that there is a specific W matrix for each edge type. And so the result of that are the aggregated information that arrive at node V from the immediate neighbors in the graph. And so that information is combined with the embedding that node V had in the previous layer, K-1, and everything is passed through a nonlinear activation function to arrive at an embedding for node V at layer K. So these constants C that you might see here are normalization constants initially. So in 2018, the state of the art for that was to simply normalize for the degree of the nodes. Today, one would use learnable weights, in particular specify an attention mechanism for that. And so then this operation is then repeated several times, large K times, where K corresponds to the number of layers. And the final embedding of the node V, Z sub V, is the embedding that is the output of the last layer. So this in the crux specifies the trip transformation function that give rise to a graph neural network that we call decagon. And so once decagon was specified as we've seen in the previous slide, we applied it to our polypharmacy knowledge graph and tested it in the way that it can answer, that decagon was able to answer queries of the following form. So if domain users or users of decagon model can ask a query whether will a form of statin and ciprofloxacin, two drugs, when taken together, will they lead to breakdown of muscle tissue? That effectively meant that in the back end, the model was asked with predicting the probability that an edge of type breakdown of muscle tissue would exist between the two drug nodes C and S that were involved in the query.
We have a couple of questions. If you don't mind, can I ask Neeraj to unmute?
Yeah, hi. I had a question. So in the polypharmacy network, like one of the tasks is like to generate paths to reason about a particular interaction between drug and a target or a disease. So do we have pathways included as a part of network in the polypharmacy network?
Yes, we have. So in particular, each of the nodes here have their own initial feature vectors that allowed us to integrate side information about drugs and proteins in the graph neural network. And for proteins in particular, we constructed binary vectors that indicate where each dimension was a particular molecular pathway, and those binary vectors indicate whether a protein is a membership of the pathway, yes, no. And those vectors were constructed for each and every proteins. For drugs, we similarly had feature vectors that represented molecular structure of drugs, or the SMILE fingerprints. And so that information would then be presented here. Those are initial embeddings for nodes would be their initial node features in decagon.
So these are not like specific entities present in the graph, but they are used to generate embeddings.
Correct.
Okay,
Correct.
Thank you.
And then we have a question from Fennel as well. Could you unmute?
Yeah. If I understand correctly, so for every layer, for every edge, we would have a different weight matrix W. Won't this be computationally very expensive to train?
No, not at all, because but that's a great comment, because if you look at this particular equation, you can see that this trainable matrix W is shared across all nodes. So all nodes use the same W matrix at a given layer, at a given edge type. So that effectively means in these knowledge graphs, and at that time, the polypharmacy knowledge graph had 1,000 edge types, meaning that we had those 1,000 W matrices to train. And that was considered to be a very highly multi-relational knowledge graph with many, many edge types, therefore many these relation matrices, but we had no problem training this model. Why is it important to have these W matrices that are specific for edge type? Because it is simply it's simply relatively naive to assume that there is a single way to aggregate neural information from neighbors of a node, independent of edge types that those nodes connect to, right? Because we cannot simply say that a given drug node might have some of its neighbors might be proteins, some of my neighbors might be other drugs to which these drugs interact by causing pneumonia, and some of the neighbors might be other drugs through which the drug interacts by causing some tumor, right? And it simply would be very naive to assume that these different kinds, very fundamentally different kinds of interactions are indistinguishable by the model, right? That's why we had to have a separate a W matrix or the parameter weight matrix for each edge type. The way you can think of it from the architectural standpoint is that essentially it means that we have a separate channel for each edge type, and then those channels are aggregated at each layer.
Yeah, that makes sense. Thank you.
There are a couple more questions, but I think we can wait until the pause at the end of this vignette.
Yes, let's do that. And so where we left off is that this approach called Decagon was trained on the polypharmacy knowledge graph. It was trained with the goal of success, with the goal of solving multi-relational ligand prediction task, which in other words, is simply means that it was trained to answer these queries of this form that, as we have seen before, that allow us to say for a pair of drug and a particular side effect, whether that pair of drug that combination of drug would lead to an adverse event that it was used in the query. So when we trained the model, we conducted a variety of different experimental tests, including different kinds of classic cross-validation settings and disease and drug-centric splits and so on. I will just mention one particular experiment here, and that was a temporal split of the data, where we used the model to, we trained the model on the polypharmacy knowledge graph that was constructed based on data generated prior to 2012. And then we asked the model to make predictions. We examined top predictions made by the model and asked how many of those predictions have been confirmed in the literature after 2012. So the top 10 predictions that the model made when trained on data prior to 2012 are shown here in this table. Recall, each prediction comes in the form of a pair of drugs and a side effect that that pair of drugs is associated with. So for example, for prediction number 8, we would read that Decagon predicted that this form of statin drug and amlodipine, when taken together, would lead to muscle inflammation. And muscle inflammation cannot be explained solely by the effects of amlodipine or this form of statin. So for many of top predictions made by Decagon, we found direct evidence for those predictions that was discovered after 2012 in the form of reports of real-world patients who were on that particular therapy and experienced adverse events that the model had predicted. And so that was very exciting and motivated us to then take those predictions that were novel, for which we couldn't find evidence in the literature, and engage in a variety of experimental validations with hospitals at Stanford and in the Boston area to validate those predictions through clinical settings. We have an interesting follow-up of this study that tries to aims to make predictions and characterizations of adverse events at the level of groups of patients rather than just broadly for any patient. And so in this particular recent study that is already on arXiv, and we have the data is already public and machine learning ready, we looked at 10 million reports of adverse events that we retrieved from and obtained from FDA. And using that data, we were able to identify what are adverse events of those drugs that are specific to certain populations of people, such as defined by age and defined by gender, as well as diseases that individuals might have. And so that has revealed some interesting inequalities in adverse events. Okay, so this concludes the first vignette. Before I move to the next one, which will be about drug repurposing, I can take a few questions. And there are many questions.
The first question is from Sun Wang, if you want to unmute. Sorry, I might be mispronouncing.
So my question is that, so currently we may not have a full picture of how a medicine functions in human body. Is it possible to use this kind of GNN to get a better understanding of use the current patterns to predict how a medicine will function?
Absolutely. So that's a great question. And the answer to that is yes. So this particular knowledge graph, the polypharmacy knowledge graph, represents current vastly incomplete understanding of biology of how drugs work. And so it was in the decagon study, the learning task and the goal was to use that knowledge to complete the graph in the sense of identifying adverse events of combinations of drugs. But there are other learning tests that are interesting to study. For example, identifying novel targets of drugs and additional targets and that are currently not known as off-target effect prediction, identifying interactions between proteins, identifying treatments that these drugs might in which drugs might be effective. And so I will mention two of those studies in the remaining two vignettes.
Thank you. And then the next question is from Rajoshi.
Hi, thanks for the great talk. So my question is regarding the interpretability of the model, or rather, how does the model explain its prediction? For example, we could give a pair of drug and it would predict few side effects. But I'm also worried about false negatives, which would be really bad, or false positives, in which case maybe those two drugs actually work together well, but if the model predicted it wrongly, we would not explore that area. So I think both of them would be really bad. So how does the model convince an end user, that is a researcher here, that their prediction is right?
Fantastic question. So I will describe that in detail in the third vignette. But briefly, we have algorithms. One of them is GNN Explainer, which were published at NeurIPS 2019, that can do exactly that and uses counterfactual reasoning to provide explanations for predictions. And in recent work that we had in collaboration with wet biologists and ex-virologists, those explanations turned out to be really crucial for these domain experts who don't have deep machine learning expertise, but are users of predictions made by these ML models for them to build trust in those predictions. So I will talk about this in the third vignette, but that's a fantastic question.
Thank you, looking forward.
And the last question we have right now is from Amaya. Feel free to unmute.
Hi. Hi, thank you for the talk. You mentioned in passing that the drug-drug interaction edges have like a likelihood associated with them. Is that used during message passing, or how is that used exactly?
Yes, so the edges between drugs have weights. Those weights indicate the prevalence of a particular side effect, meaning how often it is really observed in patients. Because it's not the case that everyone who takes a particular drug will experience side effects, right? But there is certain chance or risk associated with taking a drug or a pair of drugs. So that means that those edges between drugs have weights, and effectively in that per layer update equation that I showed a few slides ago. In the case where the neighbors of a node are drug nodes, those edges act as additional weights that are multiplied by the associated weight matrix.
I see. Okay.
That, of
Thank
course,
you.
required a bunch of data wrangling and pre-processing before the graph neural network learning, because it was really important to ensure that these estimates of prevalence or incidence of a certain side effect in a population are somewhat normalized and are on similar range, which is a major problem that the field of pharmacology is actually concerned with.
I see.
And we relied on kind of the techniques that they have developed over the last several decades, but primarily they are based on a notion called ROR, which is which is known as reporting odds ratio.
Great. So you've answered all the questions that have come up so far, and we can do more questions after the next vignette.
Great. So let's move on. And next, I will talk about a different endpoint. So in first vignette was all about drug safety, right? So that at the high level, that was the key question we want to understand and predict. And now in the second vignette, we will focus on efficacy, or whether the drugs are really effective in the sense that they achieve desired therapeutic effect. They really help patients. And so the motivating problem for this particular area of research is the process of drug development and discovery. We have seen a surge of interest in machine learning community for a variety of questions related to the development and discovery of new drugs. One of the reasons why that has happened over the last few years is, of course, because of the data availability, but also because the process of drug discovery and development is extremely resource intensive and expensive. And so it takes on average 12 to 16 years, and it can cost between $1 and $2 billion to bring a new drug to the market and design a new drug from scratch. And so that's really expensive and challenging. And so there's lots of efforts now into how to use machine learning to expedite the discovery and development of drugs. And so faced with these skyrocketing costs for developing new drugs from scratch, one of the questions that researchers are investigating is that of repurposing. So repurposing is a term that is used to denote a setting when a drug that already exists on the market, so it's currently approved, or it is in the later stage of clinical trial development, is repurposed for a new disease. So the goal is really to find whether there are certain new tricks that we could use existing drugs for. So perhaps the most famous example of repurposing is that of Viagra, which was initially designed for a cardiovascular disease, but then was repurposed for other uses. Okay, so at the core of that question of identifying therapeutic opportunities for drugs is the task of predicting what drug might treat what disease. So we can think of this problem at the high level, and we will then unpack it a bit, as a problem where we have a large bipartite graph, where one part consists of drugs. I'm showing that on the left, the other part are diseases, and we can have tens of thousands of diseases that are currently codified in medicine, and thousands or tens of thousands of drugs that are approved or experimental investigational currently in the US. So these black edges that connect drugs to diseases indicate known indications, meaning that an edge between this red drug and this disease that look like headache would indicate that the red drug is indicated, it's being prescribed, it's used to treat headache. And so our goal or learning task here will be, we want to have a model that will predict what diseases a new drug or this particular drug and its molecule might treat. So what it will be our key insight to do that? Our key insight to do that will be to really expand this view of bipartite graph from the previous slide slightly, in the sense that drugs and nodes will not be represented by individual nodes in the graph, but they will be represented as subgraphs. So our problem setup is then the following. We have the underlying graph, which is shown here as that of white nodes and black edges. That graph in practical settings will be a graph where nodes would be human proteins and edges would indicate drug interactions between them. And so on top of that graph, we overlay a large number of subgraphs representing drugs and diseases. So every disease will be represented by a subgraph of that PPI network, defined on proteins that are known to be associated with the disease. So if a certain protein, when mutated, leads to a particular disease, then that protein would be included in the subgraph for that disease. Every drug would be represented as a subgraph of the PPI network defined on the set of proteins that are targeted by the drug. So in pharmacology, there is a notion known as pharmacological space. And in pharmacology, they would say, it's accepted paradigm that a drug is likely to treat a disease if it's close to the disease in that pharmacological space. Okay, so that sounds fantastic, but what does that actually mean from computational machine learning standpoint? How can we now operationalize pharmacological space? And so our key idea will be that we will use the power of embeddings to operationalize closeness in pharmacological space. And we will learn embeddings for these subgraphs in such a way that when a drug is used to treat the disease, then the embedding of the drug will be close to the embedding of the disease. And so that will be an operation, a practical way to operationalize this notion of pharmacological space. So that means that effectively what we have previously seen as a simple link prediction on bipartite graph will now become a link prediction problem between these drug and disease subgraphs. So our input data will consist of a large number of disease subgraphs and drug subgraphs, and that are linked based on known treatment information. The method will learn embeddings for every disease and drug subgraph. I will explain how that is done. And then those embeddings will then be leveraged to predict links in novel relations between diseases and drugs, those relations representing what are other diseases that a given drug might treat. And so the main question for that method is really how to generate embeddings for subgraphs. And so for that, we recently developed a methodology known as subgraph neural networks. It has been just published at NeurIPS last year. And so subgraph neural networks, the idea of the method is that we will generate subgraph embeddings by thinking of distinct aspects of topology that subgraphs might have, in the sense of what is their position relative to the underlying graph, what is their structure of the neighborhood, what is connectivity of a particular subgraph. And so 3 distinct neural channels, which are each channel is a neural message passing network, are then aggregated to arrive at the subgraph level embedding that allows then to us to generate an embedding for every drug and disease subgraph, and then leverage those embeddings for predicting relations between drugs and diseases. So a very quick overview of SubGNN or subgraph neural network is the following. The method has 2 core parts. So the first part is that of hierarchically propagating messages in graph G. And so the idea here is that because we specify a message passing scheme that propagates messages from the underlying graph G, this graph with white nodes, to the level of subgraphs, and then that information is aggregated at the subgraph level and passed then down to the level of underlying graph. And in second part, and second important component of the model is that this routing of neural messages along edges of the graph is done in such a way that is cognizant of three aspects of subgraph topology: position, neighborhood, and structure. And so that has been very important, and we've done a variety of experiments that are described in this paper cited below to show that SubGNN can capture the relevant topology of subgraphs and can generate really powerful subgraph embeddings. We then use and apply that method to generate embeddings for diseases and drug subgraphs, and then use the learned embeddings for link prediction in the decoding phase. So to use the method for the problem of drug repurposing, we collected and constructed a large drug repurposing data set. That dataset consisted of protein interaction network for humans that was called over 50 knowledge databases. It consisted of drug protein, disease protein links to define drug subgraphs and disease subgraphs, information about known indications of drugs, meaning what are known current uses of drugs for treatments, and a variety of side information of drugs, such as membership of proteins in pathways, symptoms for diseases, structure of drugs, and so on. And that gave rise to this particular representation where we have the underlying protein interaction network, and on top of it we have a large number of disease and drug subgraphs. So we benchmarked the method by conducting a variety of different cross-validation settings, but the part of results that I will show here is one particular study that was very exciting to us. And it was a study done with the SPARC Translational Research Program at Stanford, which is an international program that has repurposed a large number of drugs over the last 2 decades. So what they gave to us was a list of drugs and diseases that were repurposed by their center recently. And so we use that list as gold standard information, and against which we evaluated performance of our subgraph embedder. And so let's look at one particular example. Let's look at the row, which is 4th row from the bottom. That 4th row says that Carvedilol was repurposed for Chagas disease by that center. And so we wanted to know, great, so if Carvedilol was repurposed for Chagas disease, then we know that Carvedilol has positive therapeutic effects for treating Chagas disease. We wanted to know how highly is Carvedilol ranked in the list of predicted drugs for Chagas disease. And so what we found, and this is in the last column here, is that in this particular case, Carvedilol was ranked as drug number 9 out of 5,000 drugs that were in the system at that time, meaning that it was ranked very highly, close to the top of prediction list. And if you look at these other ranks for other drug disease pairs, we can see that in many cases, the drugs that were indeed repurposed for the diseases were predicted highly and were ranked close to the top of the list, so of prediction lists. And so that led to exciting follow-up research that we have had on identifying repurposing opportunities for prostate cancer and schizophrenia with Stanford Medical School. Okay, so that concludes the second vignette. And now, if there are no specific questions, I will move on to the third vignette, which is that about repurposing for emerging diseases and never before seen phenomena. So let's start. So this particular work was motivated by an emerging disease, and last year that was COVID-19. And when COVID-19 occurred, there was of course a very important question, and it still is, to identify therapeutic opportunities for COVID-19. And at that point, it was realized that traditional approach of iterative development, experimental testing, clinical validation, and approval of new drugs would not be feasible. If you recall, we have seen in the previous, in the last part of the talk, that the design of new drug from scratch would take more than a decade. So that was not a feasible approach. So more realistic strategy would rely on drug repurposing and essentially tasking us to identify, to go through all the drugs that were on the market at that point in time, so early last year, and identify those that might have therapeutic effect in COVID-19 patients. So my group at Harvard was part of one task force for this particular problem, and we engaged in work that took place between mid of March 2020 and then late summer 2020. And during that time, we really compressed years of work into weeks or even days at some point in time. And that involved from collecting input data, developing and training machine learning models that generated predictions, ranked lists of drugs that might have effect in COVID, and then experimental tests and screening of those most promising predictions, as well as validations. And really to be able to do that so quickly, the very important component was that of explainability and ability that users of our ML predictions, which in our case were virologists and biologists, were able to interact with graph neural networks, receive explanations, and provide feedback to the model. Okay, so what is the core technical challenge of doing this work? Why is that not just a simple application of what we have seen in the last part of the talk? And so the part that is really challenging is because we need to make prediction for, we were asked to make prediction for new disease, for disease that we have never seen before, meaning that there was little or no information available about the disease. And so in ML, generalizing to new phenomena is typically very hard. And in the context of graph neural networks, prevailing methods still require abundant label information, whether those are examples of labeled nodes or examples of labeled links. It depends on a concrete task. However, labeled examples in many settings are scarce, and that includes the setting of emerging diseases. It also, similar problems arise if you're interested in modeling rare diseases, novel drugs in development, hard to diagnose patients, and so on and so on. So what prevailing methods assume really is that they assume that we start with graphs that are already have abundant labels. But what happens in real world, especially in these particularly new challenging problems, is that we have very few labeled examples. And so that, of course, raises a question, which is that of how to perform powerful meta-learning and few-shot learning for the case of graphs. And so let me just give a bit of background on few-shot learning for those who might be new to this particular concept, and that is independent of graph neural networks in particular. But the idea is that few-shot learning, which is a particular instantiation of meta-learning in the field of supervised learning, allows us to effectively design models that can very quickly adapt to new tasks at test time. So in particular, what I'm showing here is an example of two-shot, three-way image classification problem, where the machine learning model would be trained in such a way that it can classify images into classes by only seeing a handful of images of a certain label. So in the case of two-shot, three-way image classification, we would only have two shots, meaning two examples of each class available, and that's the information, labeled information, the amount of labeled information the model could use. So the idea in few-shot learning phase is that we have, in the case of few-shot learning, is that we have two phases, meta-training phase and meta-test phase. Meta-training phase, it's a training regime that is designed in such a way that the model is trained on these hard settings, where in each setting it has very few labeled examples of classes. And if it can be effectively trained in this way, then at test time, when it will be presented with a task that also has very few labeled examples, it will be able to generalize successfully. So the goal of this project, first from the ML standpoint, is how can we now design few-shot learning schemes for graphs? And because we had to make predictions on a completely new label set, in our case for completely new disease, COVID-19, by essentially having only a few or no examples of labels. And so to answer this question, we developed a machine learning model that is called GMeta, which was also published at last NeurIPS at the paper called Graph Meta Learning via Local Subgraphs. And what the GMeta can do, GMeta is an effective meta learner for graphs that is powerful in the sense that it can make predictions for a new label set that at test time, even though that label set was not seen during training. And the way it can do that is by specifying a large number of training and test tasks, and then training, and then addressing those tasks in kind of batch mode optimization. But the problem formulation here is that GMeta meta learner is such that it can classify an unseen label set, the blue one, by observing other label sets, yellow ones, in the same graph. So in our case, the blue label set would be COVID-19, and yellow would be other diseases that are much well understood. So the schema for training GMeta was similar, but now we didn't have images, but we have nodes and their labels. And in that particular case, we defined a variety of training tasks. Each of those training tasks had a small number of shots or labeled examples in a few classes. And because of this training strategy, at the test time, GMeta was able to very quickly make predictions for a label set that was label set 3 that was not seen during training time, where only label set 1 and 2 were seen. The key idea of GMeta to be able to do that is based on the notion of local subgraphs. So let me just very briefly explain what I mean by that. So what GMeta does, it uses local subgraphs to effectively transfer knowledge and useful information from one task to the other task. Prior work done before GMeta use a mechanism for transferring knowledge that was based on the structure of entire graphs. And so that, however, required that those prior meta learning methods had to learn graph level signature functions, which were and perform routing of neural messages across entire graphs, which is A, not scalable, and B, it also prevents prevented the models for capturing the structure of full graphs. Instead, GMeta specified subgraph level signature functions that were then effectively transferred from one task to the other. And the way that was done is that GMeta extracted small subgraphs around each labeled example, and then it applied GNNs to each subgraph individually. And that gave rise to these subgraph level signatures that were then transferred from one label set to the other label set. You might ask, okay, why these ideas of local subgraphs? So the intuition to do that is the following. We can think of graph neural networks as using two kinds of signals in graphs. One is that of label propagation, where the intuition is that nodes that have the same label are nearby in the graph. Because of that, these network message passing schemes and graph neural network architecture can effectively propagate labeled information along edges and transform it non-linearly. The other source of GNN power is of structure similarity, meaning that nodes with the same label have a similar network shape, similar structure of their local neighborhoods. Now, the motivation is that when label data are scarce, the label propagation will not be sufficient. And we need to better rely and leverage structural similarity. And structural similarity is leveraged exactly through local subgraphs, because local subgraphs capture the local structure around each and every node. And that then similarity of that local structure is then compared across tasks instead of similarity of labels, because labels are not shared across those different tasks. So just to kind of conclude here and briefly discuss results, using this algorithm GMeta and the power of few-shot learning for graphs, we then use it for the problem of repurposing on COVID-19, where the first question to ask was, well, how can we represent COVID-19? And we represented COVID-19 as the neighborhood of the human PPI network that is targeted by the virus. So the virus here on the left targets human cells and human proteins, And those proteins that are targeted by the virus would constitute a subgraph of COVID-19. So that subgraph, that neighborhood of the protein interaction network for COVID-19 actually looks like that. It's a neighborhood of 332 human proteins. And that was now an additional information about a subgraph added to our many other subgraphs for many other diseases and drugs that are well understood. And the question then, the step then was how to pre-train our meta-learning model on those drugs and diseases that are well understood, and then task the model to make predictions for this new label defined by the COVID subgraph. That resulted in embeddings for drugs and diseases, including the embeddings for COVID-19. And it allowed us to ask, well, what are nearby drugs in that learned embedding space that are embedded close to COVID-19. And that similarity in the embedding space was used to make treatment recommendations for COVID-19. Experimental results show that GNN-powered models achieve higher performance than some of the prior network medicine-based work using random walks, diffusions, or network proximity metrics. And that was calculated against clinical trial data. But then more exciting was really to take predictions made by the model and experimentally validate them in the lab. And so that is something that our collaborators at National Emerging Infectious Disease Laboratories did. And importantly, a major output of that effort is a finding of 77 drugs that showed a strong or weak effect on the virus across a broad range of concentrations. And that's an order of magnitude better than what would be possible with prior work that would rely on screening of all drugs. Okay, perhaps just a final thought is this question that was raised on how to allow domain scientists to interact with AI and ML models. And so a major part of this effort was really to implement some of the techniques that we developed earlier based on GNN explainer that can explain predictions made by these graph neural networks. And then those predictions were presented to the users and turned out to be very important for users to build and trust into the model. So I'll stop here and conclude with this observation that complex rich data interconnected data sets are transforming science and medicine, and machine learning on graphs can unlock these data sets. Most or all data sets, papers and code I mentioned today are in our lab website. You will also see we have a new initiative called Therapeutics Data Commons that has machine learning ready, contain over 60 machine learning ready data sets for machine learning in therapeutics field. So if anyone is interested in leveraging some of those data sets in their ML work, you're very much welcome to do so. Thank you.
Thank you so much for this amazing talk. Let's all unmute briefly so we can thank the speaker. Thank you so much, Professor Zitnik. Hopefully we can spare about 10 minutes for questions. The first question is from Rajeshi, if you want to go ahead and unmute.
Yeah, hi. So this is a great talk. Thank you again. So my question this time is, what is the difference between a subgraph neural network versus, let's say, just an ordinary graph convolution network or a graph neural network where during message passing, we restrict the number of hops to a local neighborhood, for example, 2 or 3. So how does subgraph neural network differ from that? And I have a follow-up question, but maybe first.
So the question, that's a great question. The answer to this is that subgraph neural network is concerned with learning embeddings for subgraphs. So not predominantly subgraph neural networks are concerned with learning embeddings for nodes, or in the case of some molecular graphs, learning embeddings for entire graphs. Subgraph neural network is designed for learning embeddings for subgraphs. And then you could say, well, of course, I can simply take my favorite node embedder and learn embeddings for nodes and take an average of node embeddings to get at a subgraph embedding. And of course, that's a valid baseline. And we show in the SubGNN paper that you can do much, much better by generating embeddings for subgraphs in such a way that we can capture a variety of topology of that subgraphs might have, both in terms of internal topology as well as border topology in the sense of how subgraphs interact with the rest of the graph.
I see. I had my follow-up question is on the third part of the talk, where you talk about graph signatures, which helps you in applying to subgraphs which were never seen during training. So how is this signature represented? Is it like in a meta-learning sense, are these like different sets of parameters which are fine-tuned at test time, or so how is this signature represented in the network?
Yes, so signatures of subgraphs are represented as embeddings of centroid nodes for those subgraphs. So the way that those subgraph level signature functions are defined is that during the process of meta training, many subgraphs are sampled across different tasks that we want to achieve generalization from one task to the other. And for each subgraph that is sampled, we generate an embedding of the centroid of that subgraph, and that is the subgraph level signature function. We show theoretically in the paper that it is sufficient for those subgraphs to be relatively small in terms of their diameter, and even if they're relatively small, they can capture most of relevant feature information, because we can show that under certain assumption, the label information decays exponentially with the number of hops from the target node. That's why then the signature functions for subgraphs are embeddings of centroids.
Right. Thank you. That makes sense. Thank you.
Great. If anyone else has questions, I think you can feel free to unmute. I think somehow our questions all got very front loaded in the middle of the talk.
I have a follow-up question on the last part of the talk again. So when you said like you generated explanations and validated experimentally, so I wanted to know if there are any general framework for generating explanations for graph neural networks in general.
Yes, let me share screen just for a moment, since I had to skip those slides, but there is a particular framework that we use that is called GNN Explainer, Generating Explanations for Graph Neural Network. We published it in 2019, and GNN Explainer, it uses counterfactual reasoning to generate local explanations for predictions. What that means is that it can explain individual specific predictions made by the GNN model. And the way it does so, for each prediction, say we would be predicting node labels and try to explain predicted labels for nodes. In this hypothetical example, it might be we have a graph where we are predicting activities of individuals that represent nodes in that graph. And if you would predict for V sub I that person would enjoy basketball, you might ask, why is that the case? And so what GNN Explainer would do, GNN Explainer defines a small optimization task that optimizes for a mask defined on a local network neighborhood around V sub I, as well as a mask on feature dimensions, in such a way that it tries to identify if certain nodes or edges are masked away, and that changes prediction made for node V sub I by a large amount, then those nodes or edges that were masked away are good counterfactuals for the prediction. So they should be put in the explanation. So that's the intuition of how we can then generate post hoc local explanations for each prediction made by GNNs. And so that then allows us, in this particular case of repurposing, that repurposing studies that domain experts were asking queries, such as will a drug treat a disease? And then the follow-up to that was, what is the predicted treatment mechanism? And so when that was asked, in the backend, GNN Explainer was run to produce an explanation, which was a part of the subgraph on which the model was trained. Around those set of target nodes that are good counterfactual examples for the prediction.
So the counterfactuals are like actual entities, or you work with the embeddings by perturbing them?
They are entities.
Okay.
They're
And
actual nodes and actual edges from the graph.
and the optimization that you do is like another surrogate model that is used to.
Right.
Okay, thank you so much.
Great. I mean, if we don't have any other questions, we can let you take a small break before your next lecture. Let's all unmute and thank the speaker again. Thank you so much. This is such an engaging talk, and it was really great for all of us to watch.
Thank you so much for the invitation. I enjoyed it very much, and I'm happy to answer any further questions via email or in our one-on-one meetings.
Wonderful. Thank you. And yeah, I think there's a different Zoom meeting, and we'll see you in there at 3 o'clock.
Thank you. Bye.
Bye.